Recording, share the screen, and there's a whiteboard. Now, Scott, can you manipulate the whiteboard? Can you write on it?
Doesn't appear so.
Okay, then something is wrong. Now try it.
I'm not given any options to do anything on the whiteboard.
Can you see the whiteboard?
I can see it. I can see your pen moving.
Okay, but you don't have the menu at the top.
Correct.
What about now? Can you see it now?
No, I cannot.
Well, I don't have any more options for allowing sharing. Let's see. So you can see the screen okay, Scott, but you just can't use the whiteboard. Is that right?
Correct. I can see the whiteboard, but I'm not allowed to change it.
Yeah, well, I don't know what else I can do. So you still don't have a pen. Can you see the menu at the top of the whiteboard?
No, I can't, and I do not have a pen.
Well, somebody just wrote on the whiteboard. Was that you?
It was not me.
Okay. Yeah, somebody's making marks on the whiteboard. So that's because I've said that all participants can use a whiteboard. So who's making blue marks on the whiteboard? Nobody admitting it?
Yeah, it's me.
Okay.
I just try because I see the like pain and then I try to do something.
Yeah, well, that's good to know. So I don't understand why you can do it, but Scott can't.
So Scott, you have to hit annotate
Where am I finding annotate?
1 second, I just lost it again. I think it's up at. I think
No,
it
I
may
found
be.
it. Yeah.
Found it? Okay.
Yeah, I just did 2 purple 8s.
Okay.
All right. Cool.
You do have access.
So yes, I can now.
Okay. All right, I'm gonna clear the board. So today, we're going to spend as much time as we need to, up to the full period, going over Mendelian inheritance problems. It's very important that you all know how to do this. The other goal is to be more engaged, have you, the students, more engaged in the class. I want to get your input on how to do these problems. Now, you should have all received from me an announcement with this worksheet Mendelian Genetics Problems, Progeny Analyses. So call that up, have that available for reference while we go through these. And Scott, you've already put up the first question, right?
Yes, sir.
Okay, well, could you continue to describe that?
Okay, so this particular problem, pretty simple one to start out with. We see that guinea pigs, they have black fur as dominant over white fur. And the question is, how could an animal breeder test whether a black guinea pig is homozygous or heterozygous? Now, this should come straight out of your lecture because Dr. Yoder did talk about this. So who wants to tell us what would be the process to determine whether a black guinea pig was homozygous or heterozygous? First off, what's homozygous mean?
So homozygous means either 2 dominant or 2 recessive genes, or what was the word?
Allele.
Alleles.
There you go. Thank you.
And heterozygous means?
1 and 1.
One dominant and 1 recessive. Okay, great. Because it's very important to understand what the terms are. Otherwise, a lot of these questions can be even more difficult to answer for you. So what is the process to determine the homozygosity or heterozygosity of this black guinea pig?
Look at that progeny and see what the traits are.
You can. You're going to do that eventually, but you need to be absolutely certain whether you're going to end up with or you're going to need to be certain why you ended up with a certain color of guinea pig. Because if black fur is dominant over white fur, what would happen if the animal, the offspring was, let's use capital B equals black fur and lowercase b equals white fur. We'll use those just arbitrarily assigned variables. But how do we, if I have an animal with black fur and black fur is dominant, what are the possible genotypes of that animal?
Can you repeat your question? I'm sorry.
So if we use capital B for black and lowercase b for white, and we know that black fur is dominant to white, what are the possible genotypes of an animal with black fur?
Would it be 2 capital B's or 1 capital B, 1 lowercase?
That's correct. So possible to be either BB or BB. And we need to know which one is, which one it is. We need to know whether this black guinea pig is homozygous, in which he'd be capital B, or heterozygous, in which he'd be capital B, lowercase b. So we need to breed this animal with a specific other animal. And does anybody remember what go ahead.
We use a chart for, I mean, PP and like PP smaller, whatever. And we use a chart.
Okay, you can use whatever variables you want. Let's. I used the big B and the little b, so let's put it in those terms.
Maybe it would help to think about the definition of a test cross, because that's what this is. And that would be, you have an unknown, an unknown that has unknown genotype, cross it with a homozygous recessive. You know what a homozygous recessive is, and in this case, it's got to be the white fur because white is recessive. It says that in the statement of the question. So a test cross is an unknown times a homozygous recessive.
So,
okay.
Yeah.
Go ahead, Scott.
No, and that's great. So if I want to know whether my animal is going to be homozygous or heterozygous, as we've said, we need to do a test cross, and the test cross is going to mean I have to cross this animal with a homozygous recessive other animal. And why will that tell me if my black guinea pig is homozygous or heterozygous? Well, if we do the cross, and let's say we have two possible options that we've already said. We can have a big B as one possibility for the black animal. And what was the other one?
Big B, little b.
Big B, little b. So this is the other possibility. And we're crossing this with a homozygous recessive, which means it's going to be little b, little b, little b, little b. Does everybody see this?
Yeah.
Okay. So we have to account for both of these possibilities. What is going to be the outcome of this first option here?
Big B, little p.
Big B, little b. And what is going to be the outcome of this one here?
Which one? The second one?
This next one down. Yeah, the bottom left.
Same thing.
Okay, correct. And now let's go to the top right. What is going to be the possibility here?
Still the same.
Still the same. In fact, they're all going to be the same. That's the only options we have when we're crossing. And what color will all these offspring be?
You say black, right?
They would be black because black is dominant. Now let's look at this right square, the right Punnett square. What am I going to get in this top left corner?
Big B.
Big B, little b, thank you. And what am I going to get in the lower left corner?
Little b, little b.
Little b, little b. What am I going to get in the top right corner?
Little b, little b.
Top right corner.
Big B, little b.
Big b.
Big B, little b, and in the bottom left corner, as we heard, little b, little b. So what are the offspring going to look like in this second case?
It's 50%.
Okay, so this is how you determine whether you started with an animal that is homozygous or heterozygous. Because when you cross them with a known homozygous recessive, if they are dominant, if they are homozygous dominant, they will be nothing but heterozygotes that are all black. So if you bred a sufficient number of these and you ended up with only black, you could assume that the parent was homozygous dominant. But if we look and we breed and the parent was a heterozygote and we breed with a recessive, we should fairly quickly see that a white offspring will appear. And as soon as we see a white offspring, we know that that one parent was a heterozygote because he carried one recessive gene for coat color. And when we cross that with the homozygous recessive, we end up with two recessive alleles and a white coat.
Yeah, so just to restate what Scott has said, when you do a test cross, there are just two possible outcomes in the F1. Either they're all going to be alike, all have the same phenotype, or they're going to be 50-50. So it's really a very nice way of determining what the zygosity of an unknown is. So is this clear to everybody? I want to emphasize that that this type of reasoning, this type of problem solving is very important. And in fact, I think the quiz that we're going to have on Wednesday will be primarily this type of problem solving questions. And certainly on the next test, maybe in the final, we're going to have problem solving of Mendelian inheritance problems. So it's important that you all be able to work these things out, as Scott did here, so that you can deal with these questions when they come up on tests. Okay, that's good, Scott. Could you go on to question number 2 and copy that onto the whiteboard?
Yes, sir, I will.
First of all, I'm going to clear the whiteboard, take everything off.
I'm typing something on it right now.
Oh, okay.
So the next part of this would then be how do you answer it? And this would just be one way of answering it. You have now seen the Punnett square logic of why this answer would be valid. But one way to answer the question, because that's what's even more important when reading the question is important, but you need to make sure you're answering the question asked. So the question is, how could you test? Well, one answer would be perform a test cross with a known homozygous recessive. If any offspring have white fur, the black parent must have been a heterozygote. And you can prove that using the Punnett squares that we just showed.
And it has to be a homozygous recessive, right?
It does, because if you had two heterozygotes, you would end up with a different proportionality. What you're looking for is really, it shouldn't probably say any offspring, but if the offspring had 50-50 white fur, if they're right around 50-50.
Okay, thanks.
Okay, so now we can clear the screen. And question number 2.
Excuse me, Scott. Now
Yes.
we know the pig should be heterozygous, right? Because they are black and white, and they are like 50-50. That's what we mean. Just we answer the question. It's heterozygous, right?
If you had approximately a 50-50 outcome, black versus white, in the F1 generation, then yes, you would be able to determine that one parent, or the black parent, was a heterozygote. Any other questions before we start number 2? All right, we have a short-stemmed yellow pod pea plant crossed with a true breeding long-stem green pod pea plant. assuming that long stems are dominant to short stems and green pods are dominant to yellow pods, we have to answer two questions. What are the genotypes and phenotypes of the F1 generation and the genotypes and the phenotypes from an F1-F1 cross? So with these questions, again, we want to look at the question itself, figure out what's important here. They tell us that short stems are the recessive, long stems are dominant, yellow is recessive, and green is dominant. Another term, what does true breeding mean? When they say something is true breeding long stem green pea pod.
Does that mean it's homozygous?
It does. It means it's homozygous because it only ever produces long stem greens. So in this case, we could say this particular plant is homozygous dominant for both traits, both for the long stem, because we're really looking at two traits here. This is what's known as a dihybrid cross, and the last one was a monohybrid cross. We were only looking at one trait. Here we're looking at two different traits, stem length and pod color. So in this case, we know because it says this is true breeding, this individual must be homozygous dominant for stem length and homozygous dominant for pod color. Again, this knowing what true breeding means, that could be a real key clue towards deciphering these questions. So let's start by assigning variables. We're looking at stem length and we're looking at color. So what do we want to define our variables as?
Any letters we use, right?
You can use any letters as long as they should make sense, but yeah, you can use any letters.
Yeah, we use, I mean, for color, I think so. We use capital C, right?
Well, we'll just use one letter for each trait. So capital C would be for what color?
For green color?
Green, because the capital is, by convention, the dominant trait. And then we would use the lowercase c for what color?
For yellow.
Yellow. Okay. Does someone else want to give me the other trait in letters?
We could do L's for long.
Okay, that sounds good. How would we set that up?
So capital L is the long stem. That's the dominant. That would be lowercase.
Okay, great. When you're doing these, it's important that you keep track which letters are which, and you can see that the big L and the little L, at least on the screen, look like a capital L and a capital I almost. So just be aware that you need to be clear with these, but I would use the same ones. And I'd use a cursive L, cursive lowercase l, because it's very different from a lowercase other letters. But these are great these are great variables. So, all right, we need to set this up. We know that what combination is going to give us long stems? Or let me ask it a different way. How would we set up the true breeding long stem green pea pod? What letters are we going to assign the genotypes for that?
Sorry, did you say short? Sorry, could you just ask that again? Sorry.
What letters will we assign as the genotype for that true breeding long stem green pod pea plant?
It would be big C and big L.
So how many alleles does each individual carry? Remember, we have pairs of chromosomes, one from the parent, one from each parent. So each one of these things will have 2 alleles for each trait. So I don't disagree with you in terms of the capital C, capital L, but do we just use one or do we have to have two letters?
It should be two.
Two, okay? So capital C, and capital L would be one way to do it. It doesn't really matter which way you go. This is the true breed long green, right? What about the short-stemmed yellow pea pod that we're gonna, or pea plant that we're gonna cross it with?
A small c with a small l, right?
Again,
How
many-
Sorry,
Go ahead.
small c, both,
okay,
and small l, small l.
small l, small l. That's good, and that is the that is correct for the short yellow. Now, does everybody see why that has to be two lower C's and two lower L's?
Yep.
Okay, if anybody doesn't, this is the time to talk about it, because another way to look at this is they didn't tell you in the question, but this is also a true breeder. This is because short stems are recessive, long stems are dominant, there cannot be a heterozygote here. It must be homozygous recessive. Same with the color. It must be homozygous recessive, because if there was if there was a dominant allele for either color or stem length, we would not display the short and the yellow. Okay, so this is all a part of how to break these questions down. Okay, so we want to cross the true breeding long green with the short yellow. And so we would lay out a Punnett square. What is the name for the offspring? What generation name do they get?
F1 generation.
So that'd be the F1, correct. So we'll just lay this out. Now, each of these plants will only pass along how many alleles to its offspring. For instance, we know that when a cell is diploid, when it creates gametes, they are haploid. So it only passes along, is it one or two of each allele to each gamete?
Should be two, I think so.
Should be two. What does haploid mean?
I mean, it's like one side of the cross, right? We should be use capital CC and LL, and capital CC and LL, and then top should be small CC with LL, right?
You're going along the right way, but this individual will pass along one of each of their alleles, one for color, one for stem length in each gamete. So we have to look at what the combinations could be from each parent. And you can use, if you remember math, algebra, you can use what they call the FOIL method.
Oh,
figure
fine.
out-
Okay.
Yeah, to figure out the possible combinations. So if we look at capital C, capital L, FOIL means firsts, outers, inners, and lasts. So
Okay.
so let's get over here. If we FOIL for CCLL, if we look at firsts, we'll get a capital C and a capital L. So you would look at this as, think of it like this. All right, break it down into each of the traits that we're looking for and the two alleles for each trait. Do you follow why we set it up this way?
Yeah, sure.
Yeah, sure.
Yeah. Yeah.
Okay. All right. If we don't understand why we're doing this, again, this is We need to talk about it now because we're looking at 2 alleles for each trait because this is a diploid individual. All right, so when these cells break down and they go haploid, we're going to get one of each letter in each gamete, but we need to make sure we get all the possible combinations. In this case, we would look at the first in each of these pairs, which would give us what? A big C and a big L, correct?
Yes.
What would the outers give us if we look at the first and the last thing, because they're the outermost letters?
The same thing.
Okay.
CL.
Inners would be,
The inner should be L.
the inside 2 letters is going to be a big C and a big L. Do you see how that works?
Can you repeat, please? Sorry, wait. Can
So
you
we did.
explain? Yeah.
Okay, so when we look at these things, we have we're trying to determine what's going to be the firsts, and those are obviously the first 2 things that we come into. Outers are what is outermost from the midpoint, where these 2 things go together. Let's see. It's really hard to. Okay, let me see if I can draw this way. So firsts would be the purple line I just drew.
That one should be the outer, right? What do you?
That, yes, that would have been outer. So
Okay,
for
the inner
draw,
should- yeah.
this would be firsts. Then
Okay.
if we look for what we would call outers, it would be those. If we use the inners, it would be these. And if we use lasts, it will
Okay.
be the reds.
Cool. Got it.
Okay?
Yeah.
This is how you would make sure that you get all the possible gamete combinations. And in
Okay.
this case, firsts give us what? Firsts are the pinkish line. What do they give
Okay.
us?
The first is CL. We got this one. Right?
Okay, so
Yeah.
let's be more exact. Is it CL or is it big C or lower C or?
Yeah, big C and big L.
Correct, big C, big L. So
Mhm.
then we look, O means outers in FOIL. So what are the outers? They're the green line.
It's big C and big L.
Okay, someone else, please. What is I? Stands for inners, and that would be the blue line.
Big C, big L.
Big C, big L. And someone else, what L is for last, and that's the red line. What would that be?
Big C, big L.
Big C, big L. Okay, so the only thing that this particular individual can pass along is capital C, capital L. So that's for the true breeding long green. Now I'm going to put up the next thing, and I would like, please, 4 individuals to give me the possible combinations for CCLL. And we'll go ahead and label that out as just for making it look the same. What would be the first gamete combination using the F, firsts?
Little C, little L.
Thank you. Someone else, the O for outers.
Little C, little L.
Inners,
Same thing.
same thing. And last.
Also same.
Okay, so what we can see is this individual can also, similar to the adult, or similar to the true breeding long, can only pass on little c, little l, and that is because this is also a true breeder. And if we cross these things, we actually don't need a big Punnett square. Because there were no additional possibilities, we really only have to look at 2 possibilities. We have little c, little l for one plant and big C, big L for another plant. I don't have to add anything to these other squares because there are no other gamete combinations possible. What am I going to get out of a plant cross or a cross here in these F1s with big C, big L cross to little c, little l? So essentially, we're going to take each letter individually and drop. We're going to take the big C and we're going to put it over here in the right. And we're going to take the little c from the top and we're going to bring it down into that same box. We're going to do the same with the L's. So what if I get a big C, little c in this first box? What am I going to get for the L's?
Big L, little L.
Big L, little l. Thank you. That is correct. Now, if you look, I took a haploid gamete from the top row. I took a haploid gamete from the left side of the column. And now I combined those. I have a diploid individual here, 2 alleles for each trait. What is the first question? What are the genotypes and phenotypes of the F1 generation? So what is my answer there? What is my genotype, first off, for the F1 generation?
The C, capital C, small, and L capital, L small.
Big C, little c, big L, little l. That is the genotype of my F1. That's correct. What am I going to get then for my phenotype for my F1s?
Green long stem and yellow short stem.
So where am I going to get how am I going to get a short, you said green long, and what was the other answer?
Yellow short.
How am I going to get a yellow short out of the big C, big L, little l? What's the dominant trait? What's the dominant trait for stems?
Should be green, right?
For stem length?
A big L.
Big L for long. That's going to be my dominant phenotype. And as you said, the dominant phenotype for color will be green. So my F1s can only be green pea pods, long stems. That would be your answer for the first one. But there's a second question now. So what do I have to do with this F1 cross? What individuals am I going to cross?
L, right?
Well, if I'm looking at individuals, so I've now bred 2 plants and I have, say, a bunch of other F1 plants. Maybe we got 50 of them, who knows? But what, I only have one option for my F1s, right? Did we get anything out of the F1 that wasn't big C, big L, little l?
No.
Okay, so then I'm going to be crossing 2 different plants that have the same genotypes because that was the only genotype I had available. Does that make sense?
Yeah, it makes sense. It's like little c and little l, right?
So I think you're jumping ahead a little bit and you're starting to talk about what gametes we're going to produce. Yeah. Okay. So if I am crossing, I'm going to have big C, big L, little l, F1. Crossed with big C, big L, little l, F1. This is what I'm trying to do now for part number 2. Does it make sense why we're doing it this way? Why this setup is the way it is?
No.
Yeah, I don't understand. I'm sorry.
Okay. The big C, big L, and the Punnett square that we just did, that came from one of the parents, correct?
Yes.
Where did the little c, little l come from?
From homozygous recessive.
Okay, from the homozygous recessive parent. And they came together and formed a diploid individual. And this individual was what?
The
What?
uppercase C, lowercase c, capital L, lowercase l.
Yes, that was the F1 progeny. How many different types of progeny did we get out of that first Punnett square? Was there one or more, or was there only one?
Only one.
Okay, so these are the only genotypes that were produced from the parental cross. And those are what's that big C, big L, little l? That is our F1 genotype. That's what we have written down here below as the answer to the first question. So do we see that?
Yeah.
Okay. So if I want to now cross, which means mate, two F1 generation plants to see what comes out, I have to take a big C little c, big L little l, which was one of my F1s, and I'm going to cross it with another F1, which is also a big C, big L little l. Did that? Do you see how I don't have any other options? My
Yeah,
F1s
yeah.
only are that particular genotype.
Got it.
Okay. So if I want to cross two F1s, I'm basically crossing two things with the exact same alleles. They're both heterozygotes in both traits, or they're heterozygous in both traits. So I need to figure out then what possible alleles each one of these can donate. And in order to do that, I'm going to use FOIL again. So let's take a look at the FOIL, and we'll break it up again as big C, big L, little l. What are the possible gametes that I can get when I use FOIL? Somebody give me what would be a possible combination for the firsts. Remember FOIL, first, outer, inner, last. So for the F, first, what would I get for a combination?
Capital
Big
C,
C,
capital
big
L.
L.
Okay, big C, big L. And go ahead, one of the two of you. What was the second? What would be the outers?
Little c, little l.
Outers would
big
be
S-
the green lines. The
it's
green
big
line in the
S-
example.
Big C, little L.
Where do the where's the green line point to? The green line in that example is the outers.
So big C, little l.
Okay, big C, little l. What about the inners?
Little c, big L.
Okay, and how about lasts? Which would be the red lines.
Little c, little l.
Okay. So those are the four possible gametes that can be produced by an F1, or at least one of them. But we can look and see that the F1s are identical, which means each plant can give the same gamete combination. So we don't actually have to work out the second one now that we've worked out the first. Does that make sense?
Yes.
Okay. And so now we need to lay out a Punnett square that gives us all the possibilities for our cross here. And that would be a 16 box Punnett square because we have 4 possible combinations for each plant. So again, when you're doing a Punnett square, one individual goes on the top and one individual goes on the side, generally the left-hand side. Don't mix up, don't try and put, you know, some of one on one side, some of the other on the other. Each individual gets its own axis. So if we look, what is going across the top, just we'll say it's that first plant, the first big C, big L, little l. What is the first gamete combination it can provide that we just worked out via FOIL?
Big C, big L.
Big C, big L. Fantastic. What's another one it can provide?
Big C, little L.
Big C, little l. Thank you. What's another one it can provide?
Little c, big L.
Little c, big L. Thank you. And what's the last one it can provide?
Little c, little l.
Little c, little l. Okay, that's one of the F1s. Now, the other F1, we'll put it on the left-hand side. And in this case, what would be one of the gametes? Well, it can do big C, big L. It can do big C, little l. It can do little c, big L. And what's the last one it can do?
Little c, little L.
Little c, little l. Okay. So now we've got both of the plants accounted for. Each gamete that each plant can provide for is along the axes. So now all we have to do is combine these. So the first thing we do, I like to go across by rows, and I start with that. So if I'm looking at the traits, what's the first letter I'm going to bring across into that top left box?
Big C.
Big C. And what am I going to bring down from the first row or the first column?
Big L.
We're looking right now at, remember we're making a diploid individual, so it has to have 2 alleles for each trait.
Big C.
Big C and big C. And how about for the next trait, the stem length?
Big L and big L.
Big L, big L. Fantastic. All right, can someone give me the complete, remember we're going across by the rows, so we're still doing big C, big L, but now we're in the next column with the big C, little l. So what would be the allele combinations I could get here?
Big C, big L, little L.
Thank you. Somebody want to do this next one? Going across again?
Big C, little c, big L, big L.
Thank you. And someone else? What's this last one in the first row going to be?
Big C, little c, big L, little l.
Okay. Does everybody see how this works?
Yes.
Okay. What I'll do is just kind of quickly try and fill these in. And let me know if you disagree with anyone I'm putting in or you don't understand why I'm putting in a particular thing.
You have a lowercase I in one of them.
Yeah. All right, I do. And that's supposed to be an L. It's supposed to be a lowercase l. I'll fix that in just a second. Thank you. Better?
Yes.
All right, thank you. And this is not uncommon. You can easily make mistakes when you're doing these things out. So the next part of the question 2 is asking us to do what?
Find the genotypes and phenotypes from an F1 and F1 cross.
Exactly.
Sorry,
ahead.
Scott, I have a question.
Yeah.
The third row, I mean, you put the capital C, little c. It should be little c, capital C, right?
The answer is it doesn't really matter, as long as they're both accounted for. And my personal preference, I can see absolutely what you're saying, but my personal preference is to always use the capital letter first. I just think it looks easier to read. As long as I'm accounting for each allele. In that case, I needed one capital C and I needed one lowercase c. As long as they're both in there, it doesn't really matter which order they go into. You just have to have this, the letters just have to match with what they're coming from. Does that make sense?
Yes, makes sense. That's a
Yeah,
good
I
point.
think that's standard genetic nomenclature, Scott.
Yeah,
That
I believe you're
correct. Do it that way.
You generally will always use the dominant allele first, regardless of where it came from, because remember, these things can combine in a variety of ways, but just for ease of reading, they typically always do the dominant allele first.
Okay, got it. Thank you.
It's a good question. Okay, so this would be your full table. What you would need to do then next would be to look at, first off, what are the genotypes? Now, some professors will actually make you figure out what all the genotypes are and how many there are of each and what percentages they are. I don't know if that's what Dr. Yoder intends or if he would be satisfied with seeing a table filled out like this.
Yeah, the latter, along with the phenotypes.
Yes, and that's the second part of the question. So for genotypes, he would be perfectly satisfied to see a table like this and it could be left that way. But then what you do have to do is take a look at what each of these different phenotypes would be and tally them up. For instance, what would be the phenotype in the top left corner? Big C, big C, big C, big C, big L, big L. What would be the phenotype?
Green long stem.
Okay, so he would expect you to do something like this. He would say green long, and you would have to start making little tick marks. And every time you came across one that was green long, you would add a tick mark there. And at the end, you would just count up all your tick marks, and then you would give him a number. So what is going to be, say, let's look at the third column, the one that has the big C, little c, big L at the top. Go down to where it's crossed with the little c, big L, and we get little c, big L, big L. What is that phenotype?
Yellow long stem.
Yellow long. So that would be different than what we got at first, so we'd have to add that as a new option. And what you would do, what you would find when you totaled all these things up is that you're going to have basically a green long and a yellow long and a green short and a yellow short. And in Mendelian genetics, when you're crossing like this, you will find a very particular numeric pattern of 9 to 3 to 3 to 1. So if you totaled all those up, that's what you're going to end up finding you have as your genotypic ratio. It will not be your phenotypic ratio, but it will be your genotypic. He's not asking you for calculating all the genotype or counting up all the genotypes. That gets confusing, but it can be done. But this is good to know. 9 to 3 to 3 to 1, that is your typical Mendelian dihybrid breakdown of phenotypes. You'll continually see those numbers when you're doing these heterozygotes in both traits. Does everybody understand how to do this second question? It's one of the more involved ones in here.
It's important that you all understand the steps, how you think through this and come up with the answers that Scott has laid out. Something like this will probably come up on a test sometime along with other types of genetic questions, but a dihybrid cross is a pretty standard procedure in genetics. So you need to know how to do this in order to deal with the test. So if there's any questions, anything you don't understand, ask Scott now.
I would also recommend before Dr. Yoder has an opportunity to blank the screens, use your phones. Take a picture of this, because you should be able to then go back to your worksheets if you didn't get this and start with a blank worksheet. Now you know the basic procedure, work through it, then compare your answer to this screenshot and you'll see whether you did it correctly or not. If there's no questions, take your pictures quick and we'll work with number 3.
Yeah, number 3 is a sex linked, and that's it's different and it's interesting.
Typically, I would say on an exam, you can expect to see one straight up monohybrid cross. Maybe it's a test cross, maybe it's just a straight monohybrid cross, but you should expect to see at least one monohybrid. You probably will see 1 or 2 dihybrids, and typically you'll see 1 or 2 sex linked. Just depends, but those are the 3 main types of problems you would be working out. All right, let's go ahead and clear this off and we'll put in question number 3. And what's good about getting question number 3 knocked out is the rest of these, because we probably won't get to them all, but the rest of these are all just variants then of the monohybrid you saw, this dihybrid, and the next one, the sex linked. All right, question number 3.
Scott, I hope you can do number 3, and then I hope you can get to number 5, because that's a back cross.
Number 5, okay. So what I'll end up doing here then is probably going through this maybe a little quicker. If you have questions on what I'm doing here, so basically I'll just do this one. And if you have questions on this, please use the discussion board. Please come to workshops where I can actually spend the time and work all these out for you. But for the sake of getting this going, I'm going to just work this out. So in humans, hemophilia, the bleeder's disease, is a recessive X-linked trait. Use a Punnett square to show the possible genotypes of offspring of a man who is a hemophiliac and a normal but heterozygous woman, where X contains the normal clotting gene and X asterisk contains the gene for hemophilia. Well, we know that by reading this, the man has hemophilia. Men have an XY chromosome, but if he's got, this is an X-linked trait, so this is found on the X chromosome. If he's got hemophilia, he must be X asterisk Y. That's the man. The female, it says that she is heterozygous, meaning she has, because a woman has 2 X chromosomes, she has 1 normal and 1 recessive allele, meaning she's big X asterisk. I would lay out a Punnett square real quickly, and I would just do a quick cross. We're only looking at really one thing here, so this is just it's kind of a variant of.
Would it not be a little x for the heterozygous?
No, because a woman truly has 2 chromosomes that are X. We would typically either use a subscripted, if you're using letter, or superscripted, I'm sorry, you would use a superscripted capital. So you could have, say, if capital H meant no hemophilia and lowercase h meant has hemophilia, for a woman who is heterozygous, you would have this X with a superscripted capital H and an X with a superscripted lowercase h. So again, the X is just the chromosome we're looking for. These are just slightly different than what we were doing earlier, which is why it's good to cover them, because they can be a little confusing.
Now that's something different about sex-linked, is that you don't have uppercase and lowercase with the sex-linked chromosome, or with the sex chromosomes.
So what individual have I put on that top row?
The female.
The female. And remember I said we don't mix the rows. One individual goes on one axis, the other goes on the second axis. So crossing this top left, I get big X, and that is going to be a normal female. Crossing the next one to the right, I'm going to get X and X asterisk. This is going to be a female who is a carrier, but not afflicted with the disease. In order to be afflicted, she would have to have 2 asterisks, X asterisk, X asterisk. She doesn't have that, so she's just a carrier. In the next row, oh, what did I do wrong here?
Forgot the asterisk.
Yeah, I forgot the asterisk on the male. This is where you can make your mistakes. So that's what the male should look like, which means that this next box would be
She have it.
Would she have it?
Yeah, it should be. She have hemophilia, because she got like 2x right.
So what I get is, in the first box, I'm going to get X and X asterisk. This is going to be your heterozygous carrier. She won't have the disease, but she will carry the trait. The second box, you're right. This will be a female with hemophilia, because she got a recessive X from the father and she got a recessive X from the mother. And in the bottom left, I get a normal male. And in the bottom right, I get a male with hemophilia. All right. That's how you would work this out. They don't ask you to explain anything else. Just show this. According to this, just says use a Punnett square to show the possible combinations of genotype. There they are. If they asked you phenotypes, then you would label,
Scott, just the fourth one. What should be, he have it or just he carry it?
In the fourth one, no, he has it because he has no other X chromosome to mask the symptom. So with recessive X-linked traits, as long as you have one non-recessive chromosome on the X, you would be okay. That's why the female in the top left does not have the disease, but she's a carrier. She has one normal X and one recessive X. In order to have this disease, you have to have 2 recessive X's, or in the case of a male that does not have an additional X, just having the single X means that they have, the recessive X means that they have the disease.
Okay, got it. Thank you.
Okay. All right, take your pictures real quick. And I'm clearing the screen now. All right. Question 5.
Can you put it back just a little bit?
I cannot. It is gone. Let's see. This is what we want to know. We have tomatoes. Red fruit is dominant to yellow. Suppose you have a tomato plant homozygous for red, crossed with one that's homozygous for yellow. Determine the phenotype and genotype of the F1, the offspring of a cross between F1 back to the red parent, which is a back cross, and the offspring of an F1 back to the yellow parent, which is a different back cross. All right. We need to label what we want as our traits. This is still a monohybrid. We're only looking at color. So I'm going to use capital R equals red, lowercase r equals yellow. If I have a tomato plant that's homozygous for red, the red plant must be big R, big R. Yellow plant must be little r, little r. This is how I would lay out my variables. For the sake of time, you can lay out a Punnett square with big R on one axis, little r on the other, but the product of 2 homozygous individuals, one dominant, one recessive, will always be heterozygotes, 100%. So what I'm going to end up with is
This big R, little r.
big R, little r for every one of the F1s. That's the only thing that they'll produce. My F1 generation will all be heterozygous. So now it asks me to take that F1 and cross it back to the red parent. So now what I've got is a Punnett square, and we'll lay this one out, but it's a Punnett square where I am taking my F1 generation and I'm going to cross it back to the parent, because that's legal in the plant world and animal world, not so much in the human world. So we're going to take, what did I get for my F1s?
This big R, little r.
Big R, little r. So because this is a monohybrid, I'm only looking at one trait or one allele per box here. And the red parent was big R and big R, correct?
Yes.
Okay. My answer here in the first top left is going to be big R, big R. My answer in the top right is going to be big R, little r. In the bottom left, I'm going to have big R, big R. And in the bottom right, I'm going to have big R, little r. So this is what I would get with a back cross. Okay. So the phenotype and genotype of the back cross F1 to a red parent, I would get 50% big R, 50% big R, little r. Those are my genotypes. I would also get what as far as color? It would be 100% red, because red is dominant to yellow. So the heterozygotes, the big R, little r's, are going to come out as red. Now, if I do my second cross, my second part of it here, which is crossing my F1 back to the yellow parent, on the top, which is going to be my F1, just because I said so, I'm going to have big R, little r, and on the side, what am I going to have? What's the yellow parent?
It's little r.
Little r,
Little r.
little r. Fantastic. Now I'm going to cross, and I'm going to get big R, little r in the top left. And I'm going to get the same thing in the bottom left. And in the top right, I'm going to get little r. In the bottom right, I'm going to get little r. So now what are my genotypes for the second problem we worked out?
It's still the same, right? No. It's 1, 2.
So what phenotypes do I get? Or I'm sorry,
It's still-
genotypes.
It's still, I think so. It's 50%, right? We get
Sure, we get 50%.
R, capital R, little r, and little r.
So we get 50% heterozygotes and 50% homozygous recessive. And yes, 50% big R, 50% little r. That's my genotype. Now, what is the phenotype going to be?
Yellow, right?
Let's take a look.
Okay, just a second.
What's the question say?
R. It's the F1 black yellow parents. R yellow.
So what color is dominant?
Oh, it's red. Sorry.
So all I have to have is one copy of a dominant allele, and my color will be red. So the first column is both big R, little r. Those are going to be red. So 50% of my offspring will be red. How about the second column?
It's yellow.
They're going to be yellow. So I'm going to have 50% red, 50% yellow. All right. That's how this works out. So my answer over on this right hand or left hand side is 100% red phenotype and 50% big R, 50% big R, little r. That would be my answer for the first question. My answer for the second would be 50% red, 50% yellow, and 50% big R, 50% little r. That would be my answer for the second question. Well, B and C.
Good. Thank you, Scott. Good job explaining these things. We're out of time. So I just wanted to say a couple of things. On Wednesday, we'll have a quiz. Obviously, we're not going to have 10 questions on Mendelian inheritance. We'll give you a couple, and you'll have all day to get your quiz done. Have 20 minutes to answer whatever questions we put there. The other thing is I will send you the same set of questions with the answers filled in so that you can review the questions, and as Scott said, read them carefully, and you can see the answers to help you prepare for future tests. Now, the reason I wanted this question number 5 done is because it is a back cross, and that's a term that I don't think you've encountered before. But back cross is the result of a progeny crossed to one of the parents. And so you can back cross as many generations as you like. And for doing inbreeding, that's very often what happens. You just keep back crossing children to a parent until everything is homozygous. All loci are homozygous. So we are out of time. Any quick questions before we shut things down?
Yes, sir. I have a question.
You're welcome, Melody.
Thank you, Scott, first. And
Welcome.
Mr. User, I just have a question. If you can, like, make it the quiz, make it for 30 minutes, if you can.
Okay, depends on how many questions we put in and the degree of difficulty, but yeah, I acknowledge that to work these things out does take time. So I'll give you more time for the inheritance questions.
Thanks, sir. We appreciate that.
So I would just again like to plug, if this was still confusing to you, or if you go through that worksheet, and because some of these questions you really have to read them. You have to figure out, they're logic questions. You have to figure out what the question is really asking you so that you know what the setup is. So if you're still having difficulty with that, please come to the workshops, or another alternative is use the discussion boards and ask questions, and I will do my best to answer them for you. Usually it's a little easier when it's all interactive and we're working together, but I will do what I can on a discussion board too.
Okay, we'll have you'll have a quiz that will not be part of the lecture period on Wednesday. I will start on the next topic, which is enzymes and energy. That's what we'll talk about on Wednesday in class, and you'll have Wednesday to do a quiz. Everybody okay? If so, we'll take down the whiteboard.
Do you want to chit chat for a minute, but when everybody leaves?
Yeah.
Okay.
All right, sir. You have a great day, guys.
Okay.
Thank you.
Take care, everybody.
Thank you, sir. You too.